Hi everyone, my name is Harleen, and for my group project, it's done by myself, Takoya, Samantha, and Ebony. For our project, we decided to focus on prion disease, more specifically the consequences and mutations when normal turns to fatal. I will be going over our abstract and introduction. The function of prions has many unknowns. Prions are transmembrane proteins mostly located in the brain. When functioning correctly, they contribute to our brain health. When mutations or disease occur, it can lead these to build up within the cell and lead to death. There are two kinds of prion disease, one which is caused by genetic mutation, called familial prion disease, and the other is acquired through infection. While little is known about the acquired versions of this disease, advancements in genetics have begun to unravel the mysterious nature of prions and genetic prion disease. Current research suggests that genomic variation in the PRNP gene, which encodes the prion protein, may lead to mutations that increase transmission and progression of prion disease. The three most common types of inherited prion diseases are Creutzfeldt-Jakob disease, Gertzmann-Straussler-Scheinker syndrome, and fatal familial insomnia. By comparing these to the normal prion protein, the source of the misfolding can be identified. Prion diseases are a group of rare progressive neurodegenerative disorders characterized by the accumulation of abnormally folded prion proteins, which disrupt cellular function and lead to tissue damage. The prion protein plays a critical role in normal cellular function, such as cell signaling, neuroprotection, and synaptic regulation. Mutations in the prion protein gene, or PRNP, can lead to serious problems, eventually causing death, while mutations in the prion protein gene, PRNP, have been found in some familial prion disease. The exact mechanisms by which these mutations influence prion protein misfolding are still misunderstood. This hypothesis proposes that mutations in the prion gene that alter the amino acid sequence of the prion protein led to its misfolding, which in return results in the loss of its normal biological function as measured by impaired cellular prion protein interactions. However, the null hypothesis remains that mutations in the prion gene do not alter the amino acid sequence of the prion protein gene and do not cause misfolding or loss of biological function. Now I'll be handing it off to my next group member who will be going over our methodology.
The methodology we used was to search the NCBI database for the Homo sapien prion protein. We then identified variants that could cause disease and a loss of protein function. We did this by comparing contigs and looking for mutations that would result in a change in translation. We then used this information to search the RCSP protein databank to take a closer look at the protein expression and the change in protein folding.
Results. Our results from the blast of the major prion protein, which is the normal prion that we find on the surface of human cells, generated several variants of the abnormal prion that cause prion diseases. We decided to look at the amino acid sequences of variants 2, 3, and 4, and if you take a look at the provided sequences, they all show a slight variation in their identities in the 99th percentile, and this is because of the insertions in their amino acid sequences at different fragments when compared to the parent chain. These insertions are identified in the results by a dashed line that is red, and that is where what we would call a misfold happened, or a disruption of the normal sequence. We also wanted to see structurally what this change would look like in different mutations of the prion disease, and if you zoom into the different mutations that we have on the left side, compared to the normal prion, the mutations show that they aggregate into long fibrils that disrupt the normal function of the prion. You can see it very distinctly in the fourth example that we have of the M166C/E221C mutation. You can see those dark blue/purple colored fibrils branching out from where that misfold happened in the prion.
Hello, this is Ebony Smith to provide our conclusion and discussion. Our findings suggest genetic mutations result in structural changes in the form of misfolding and dysfunction of the prion protein. The M16V and M16C/E221C prion protein variants specifically stand out as they are associated with inherited diseases as they exhibit changes in their amino acid sequences. Utilization of both sequence alignment via NCBI BLAST and structural visualization via RCSB protein data bank. This study provides insights to alterations at a molecular level that help explain how mutations in the PRNP gene lead to the progression of the prion disease. Further analysis is essential to understanding the full extent of how these mutations contribute to the disease. Future areas of exploration include acquired forms of the disease, understanding the mechanisms how prion proteins misfold, and how they interact with other cellular components. Therapeutic strategies may even result from understanding the function and stability of prion proteins. This concludes our presentation, and thank you for your time.